Okay, so I'm going to get started on I guess, a three-part lecture on behavior genetics. The first one will be quite brief, and I guess just introduce a notion of heritability and what it means, what it doesn't mean, and eventually we'll get to how you actually estimate it using behavior genetic methods. So let's start with kind of informal definition. And most of you know this already. Dan already talked about what we call the genetic factor and how we can decompose it into an additive part and then another part. And so the first thing to do when we introduce the notion of heritability is to make a distinction between what's called broad and narrow heritability. So broad heritability is about the variance of this genetic factor as a whole, so the best predictor, relative to the phenotypic variance. Narrow heritability is about the variance of the additive component of the genetic factor. Okay? And there are good reasons to care about both of these things. It really depends on what your research question is. Later on in the course, we'll also talk about other notions of heritability, such as SNP heritability, and that refers to the proportion of variance explained by some SNPs that you've genotyped. They're genotyped on your array. Oh, okay. Now I've lost control of the. What do I need to do? Okay. So, yeah. So recall the model from the causal model from Dan's lecture. We had this gx object. We had this vector z that Dan will talk about a lot more, especially when we get to GWAS. We have some residual, and we can decompose. We can always decompose gx into an additive and a non-additive part. So broad heritability then is the variance of g of x over the variance of y. And we can take Dan's causal model and we can just rearrange it so that we put the non-additive part into the residual, so that now we have the additive part plus the covariates plus the residual that we redefine so that it absorbs the non-additive part. With that alternative formulation, we define narrow heritability then as the fraction of this ax object over the variance of a. Okay. So in practice, okay. So a couple of caveats before we begin. These heritabilities, they're like any other descriptive statistic that you calculate for a population. There's no reason a priori to think that it would be, some universal constant. So they're population specific and they can vary over time and across environments. The second thing is sometimes we will talk about the heritability and the R-square of a regression almost interchangeably, okay? But the way we defined it, I mean, when we do that, we're sort of implicitly making an assumption about that this AX is independent of the other stuff, right? Because otherwise the variance of AX over AY is not the same as the explanatory power that you would get if you just regressed Y on this AX. You're going to get some additional predictive power if your AX is correlated with Z and stuff that maybe goes in the residual, though that's probably not a concern. So it can be helpful to think of the narrow heritability as the R-squared from a population regression, but realize that when you do that, you're kind of making an additional assumption, most likely. And the reason that this problem of covariance between AX and other things is the reason that I don't think it was a history could have turned out differently. It could have been that we could have ended up with a definition of heritability that was like a standardized regression coefficient. I think that's what Wright wanted, and then we wouldn't have had this problem. Okay, and some of the other issues that we'll talk about when it comes to the misinterpreting proportions of variance and what they mean. Okay, now there are some good reasons to focus more often on narrow than broad heritability. For the purpose of this course, a big one is going to be tractability. I mean, GX could just be an incredibly complicated object, right? So remember, as Dan explained, GX includes potentially all these higher order interactions between dominance and additive effects of individual SNPs, right? And those are just extremely difficult to estimate credibly, without enormous samples, much bigger than what we have right now. So, as an empirical matter, it's often useful to start by studying AX. The second is that sometimes narrow heritability is just of greater theoretical interest. I don't know that that's very relevant for what we'll be doing here, but in animal breeding, for example, it's a key parameter because it actually tells you something about their response to selection. And then the last thing is that there's good prior evidence and also some theory suggesting that we should often find that the variance of this, the ratio of, say, narrow heritability to broad heritability is often a fairly high number. Okay, so those are the three reasons. Okay, now on the problem set, we gave you, we asked you to work through a simple single locus model. I mean, it was simple in the sense that it was all elementary statistics, but it was not simple in the sense that it's extremely, I think, tedious and easy to go wrong. You know, one line of the calculation, you make a mistake, and headache ensues. But I just want to make a couple of remarks about it. And so the reason we've had you take a look at a single locus model is that a lot of the insights from the single locus model, for example, about the resemblance in different types of relatives, kind of generalize to multiple loci, and that's going to be a really important point that we'll talk about at length. And the second reason is I want to try to clarify some terminological issues that I was confused by a very long time. I hope I understand it now. And I think it boils down to, a lot of it boils down to the failure to clearly distinguish what we call dominance deviations, the dominance parameter, little d, and the proportion of variance explained by dominance deviations. So here's some figures from the Lynch and Walsh textbook, just showing. Well, on the left we have the purely additive model. So this is the BLP, and this is the case where the heterozygote is just the midpoint of the two homozygotes. So there's no dominance. The dominance parameter is 0, so little d. Here is an example. Here's an example where we varied. We increased the dominance parameter a bit, and we get some new best fitting regression line. And a key thing to realize is that the slope of this line depends not just on these 3 points, but also the number of observations in each point. So it's a regression slope, right? You're not running a regression with 3 data points. And here's a third case, it's known as overdominance, and I think, but in GWAS we almost never see anything like that. Okay, so the point is just that the BLP will vary predictably based on the dominance parameter, but not necessarily in a way that you might think at first. I'll have more to say about that. The other thing you can ask is what happens if we vary the allele frequency? So we push more people away from homozygosity to being heterozygous, and some of the heterozygotes towards being homozygous. And so as we progressively increase the frequency of the reference allele here, what happens is that the best linear predictor, so the regression slope, attaches much more weight to being close to these 2 data points, because that's where we have the most observation, right? And regression just minimizes some squared deviation between the predicted value and the actual genotypic value. So the slope will change if we vary the allele frequency, and so will the dominance deviation. So the dominance deviations are the difference between the genotype value, like the actual expected phenotype for somebody with each allele count, and the predicted one, right? So here the dominance deviation would be this difference, and here it would be equal to this difference. Okay, and so hopefully those graphs help a little bit, clarify some, I think, what are common mistaken intuitions. One is that increasing z necessarily reduces the variance of explained by additive genetic effects. Okay. On the next slide, I'll give you, I'll show a little bit more about why that's not necessarily the case. The second is that if this d is large, then it's surely the case that the variance of d of x is large relative to the variance of the additive factor. And I think, in reality, the thing to realize is that the dominance parameter, the dominance deviation, and this variance of the dominance deviations are quite different objects. And I think on the homework you derived something like this. You derive the formula for the additive genetic variance, okay, where this second term is the squared slope of the regression slope coefficient from the regression, and you derive the second term for the variance of dx. And from these equations, it's what happened now? From these equations, it's fairly
No, it's my fault.
it's fairly immediate that increasing d, for example, doesn't necessarily mean that the variance of a has to go down. And the reason is that the sign of the derivative just depends on q minus p, the sign of q minus p. That's the same thing. You can also come up with
Do you have control? Or?
Yeah. And it's also easy to just find numbers such that d is a very large number, but most of the the narrow heritability, the ratio of the narrow heritability to the broad heritability is something close to 1. Yeah. So, okay, so now that we've talked a little bit about what heritability is and some issues that immediately arise when we try to define it, I just want to walk through two common fallacies that show up. They're quite common when you talk about this parameter with laymen, say. Okay, and I want to distinguish between two fallacies, one of which I call the Goldberger fallacy, the second of which I call the Jenks fallacy. And the Goldberger fallacy is the notion that the heritability is some sort of index of policy effectiveness. If the heritability is high, it means that the phenotype can't be changed through environmental interventions, and maybe vice versa. And it's a common error, and I'll try to illustrate it with a famous example from the 70s. So Taubman and co-authors had published a paper in the AER that tried to estimate the heritability of income using data on twins. And asked to comment to the Times of London. Hans Eysenck was a very prominent differential psychologist in London, offered the following interpretation of the findings. So these results really tell the Royal Commission on the Distribution of Income and Wealth that they might as well pack up. Okay? So Goldberger's retort has become very famous. And I should say, Goldberger was not always the most polite in his writing, but he just felt very strongly that there was a lot of abuse of behavior genetics, and I think it shows. Okay, but I'll read it aloud. So powerful intellect was at work, that's referring to Eysenck. In the same vein, if it was shown that a large proportion of the variance in eyesight was due to genetic causes, then the Royal Commission on the Distribution of Eyeglasses might as well pack up. And if it was shown that most of the variation in rainfall is due to natural causes, then the Royal Commission on the Distribution of Umbrellas could pack up too. So he's pointing out sort of two problems with Eysenck's claim. The first is sort of confusing the proportion of variance explained by some variable within a sample with the effectiveness of some environmental intervention that potentially introduces a completely new environment. And the example he's giving is eyeglasses. It's entirely conceivable that before eyeglasses have been invented, eyesight, like everything else, is going to have some heritability, and maybe that heritability is really high. It doesn't mean that giving eyeglasses to people is something that can't possibly pass some sort of reasonable cost-benefit test, right? And the second issue is that if something is genetic, something is associated with genes, then we shouldn't try to do anything about it because it's quote unquote natural, and that too is a kind of fallacy, I think. I mean, there are plenty of diseases that have natural causes in the sense that they have some some genetic basis that we can trace. It doesn't mean we shouldn't try to cure cancer, right? So it's just an entirely separate issue. I sometimes call it a naturalistic fallacy, I think. So that's Goldberger. And the second fallacy is what we call Jenks's fallacy. So Jenks has a very famous, great paper, in my opinion, in I think the American Journal of Sociology, that laid out, that explained why a high heritability doesn't mean that environmental factors don't explain a lot of variance in the trait. Okay? And the reason he gave, which is a good one, is that genes can often influence outcomes through channels that we would think of as environmental. I'll give some examples in a moment. But a good illustration of this is, and he gives some of his examples in the paper, actually, in the spirit. A good illustration of this is how, like, just a change in attitudes towards women in higher education. So when my grandmother wanted to go to high school, she was born in 1928, her father thought she was out of her mind, and that was probably the predominant view at the time. And now you go to Iceland, and a majority of college students are female. So there's just been a huge shift in norms. And there's obviously no question that just a few generations ago, there were just enormous barriers that you faced if you had two X chromosomes and wanted to pursue higher education, and that, in a large part have been torn down. And so, that's an example of how something, quote unquote, genetic, having two X chromosomes, evokes a certain environmental response that can vary quite dramatically over time and space. But for reasons we'll talk about, those kinds of effects in the behavior genetic variance decomposition will often show up. Well, will show up as will be part of the heritable variation. We'll talk more about that. And the other thing that the other mechanism, a causal pathway from genes to some outcome could run via selection into certain environments. So, for that reason, thinking about describing the error term as environmental is fine. I mean, I'm sure I do it sometimes, but you have to, if you use that language, you have to make it clear that you're not necessarily implying that the other terms, like the genetic factors, are non-environmental, because genes could influence the outcome through the environment. And one way to think about it, there's like a distinction between what we mean by environmental effect in common parlance and what it means in the behavior genetic decomposition. So let me illustrate with two examples. The first one is from a paper on obesity that starts by juxtaposing genetic explanations for obesity with things like unhealthy lifestyle choices or environmental or other environmental factors. Okay, and so what's the potential problem? The potential problem is that these things need not be mutually exclusive. It could be that some of the reasons that you find that genes are associated with risk for obesity is that some genes influence things that they would think of as lifestyle factors, diet, exercise, these sorts of things. And if there are genetic effects on those lifestyle factors, they would show up as SNP associations with individual genes at some level, if you have the right data and you do the right analysis. So you'd find that, and it would be an effect of genes, but it would be an effect of genes that was operating through environmental channels. Okay, so if you're interested in factors that influence obesity risk net of things that you consider environmental, then you face the difficult challenge of sort of trying to find associations conditional on all the environmental factors that you want to rule out as possible explanation. And here's a second example. And I have to say, so this is Andrew Yang. He concludes, his conclusion is fairly uncontroversial. We should build bridges and be nice to each other, yada yada. But the premise of this conclusion has very little to do with the conclusion itself, and that's all I want to say. And I don't mean to pick on Andrew Yang. He's probably the only politician who's ever heard of a twin study. So credit to him. But and I do think that he here is making a common error of judgment. I guess his implicit thinking is yes, if something is heritable, it has to be hardwired, and therefore we should just respect each other because there's nothing we can do about each other's views. That may or may not be true. That has very little to do with whether attitudes are heritable. Okay. So let's just work through a silly example of the Jenks critique to illustrate the point about environments and genetic influences not being environmental and genetic influences not being mutually exclusive or not necessarily something to 100%. Here it is. So my BMI is going to be determined by my diet, a bunch of other heritable traits, D, and some other stuff. Okay. So we can say this is a sense in which we can say that the fractional variance in BMI due to environmental factors is 100%. Like, by assumption, all the stuff in D is things that we think of as environmental. Okay. Now suppose that my diet and these other things are influenced by some additive genetic factors. So you just substitute that second equation into the first and rearrange, and what you get is some heritability that depends on this gamma parameter, the variance of xb, and over the variance of bmi. So what's the point of this example? The point of the example is that if you calculate the fraction of variance in bmi, due to the environmental plus genetic factors, you get a number greater than 100%, because we set it up so that all the genetic effects were operating through the environment. Okay, let me check this out. All right, so maybe I'll go through this slide, and that might be a good time to break, because then we're moving to the next topic, so to speak. So why then should we care about heritability if they don't tell us anything about policy, and if they don't tell us anything necessarily about the fraction of variance that's explained by environmental factors? Well, a couple of reasons. One is that they're sort of facts that should constrain the set of theories that you consider plausible about the world. For example, holding everything else constant, a higher heritability means you should be more concerned about genetic confounding when you're trying to estimate some environmental effects, say. So, common one is parental income on children's outcomes. And towards the end of the lecture series, I'll give an example that's that illustrates how you can answer the question of whether parental income is causally related to children's outcomes with a little bit more credibly by using adoption data. Okay? And so the idea there is that at least you've eliminated one source of correlation between parental income and children's outcomes. Of course, we know that if we look at children who were reared by their biological parents, there's a pretty strong income outcome gradient for most outcomes that we care about, things like test scores. And the question is, what explains this gradient? Is it a causal effect of income per se, or is it some unobserved third factor that's causing them both? Or maybe it's more realistically some mix of these two things. And so just being able to rule out the genetic confounds, or at least argue that you found a setting where they're not likely to matter very much, is a step towards answering this question more convincingly. The other reason to care about heritabilities is that they tell you something about how accurately you could, in principle, predict somebody's outcome from their X's. Okay, and as we'll see throughout this course, the predictive power of these scores or indices, the different fields use different labels, is increasingly both practical and feasible. And a second reason is that heritability is kind of a necessary condition for gene discovery, and all else equal, you probably want to, if you have a choice of doing a GWAS of either a phenotype with a modest heritability and one with a higher heritability, then all else equal, you probably want to go for the one with the higher heritability. You're also finding something that are greater. Okay, so I think that's a good way to pause before we start talking about BG and the basic approach. Okay.
Sure.
Yeah.
Okay, so we're going to stop now for lunch. So David's going to continue.
Okay. So we talked about what heritabilities are. We talked about some common misperceptions of what they mean. And we talked about some ways in which they might be useful. So now I want to turn slowly towards the question of how do we estimate them, and I'll talk mostly about traditional behavior genetic approaches, which fundamentally are about looking at the resemblance of different types of relatives phenotypically, and trying to then make inferences about these latent variables, including latent genetic factors, based on some sort of restrictions that you get from theory. But also intersperse, I think I'll throw in some thoughts on how, along the way, on how GWASs might be helping us identify these models more credibly, specify them more credibly, is helping us kind of discriminate between alternative theories that, before we had the GWAS data, we couldn't really credibly distinguish between alternative explanations. And as a result, what ended up happening in behavior genetics is that people formulated very different models that could both, quote unquote, explain certain features of the data, and it was just hard with the available information to discriminate between them in any serious way. Okay, so it's useful to begin to just take our our G and our U and standardize them so that they have mean zero and unit variance. And so then we can write our causal model like this, where now G is a coefficient that tells us the effect of a standard deviation increase of G on Y. We haven't necessarily standardized Y yet, and likewise for U. Okay? And now, suppose we want to know something about these G's or these U's, and the problem is, of course, that we only observe Y's. Okay, so at its core, behavior genetics proceeds by saying, okay, let's, and I'm gonna just use a prime here to denote the second individual in some pair of relatives. Okay. Let's separate. Let's identify different types of relatives, different kinships indexed by K here, and let's calculate the phenotypic covariance between individual 1 and individual 2 in some pair, and the order is arbitrary. And under some assumptions that covariance is going, you can write it as a genetic correlation times G square, where G square is the variance in this genetic factor. This rho Ku times U square, where U square is now the variance in the environmental factor, and a covariance term. Okay. And the BG approach then boils down to measuring phenotypic resemblance of kinships who vary. Either we know or we think we know something about how this value of rho G varies across individuals, or we might have something to say about the extent to which they share environments. So typically, the choice of rho Kg comes from genetic theory, and then what people do is try to leverage separation experiments to identify types of relatives that typically grew up in separated environments, and then use that to justify some restriction. Typically, that these covariances are zero, though in practice we can think about what that means, and you can imagine other ways to proceed. But the kind of textbook example is you find some sibling types who were separated, who were reared together. You find some other siblings of the same genetic relationship who were reared apart, and you assume that the difference tells us something about the extent to which the reared together siblings share their environments. Okay, but we'll see many examples of this as we proceed. What I want to emphasize as we get started is like the distinction between the ideal experiment that we'd want to run here, maybe to try to infer these parameters, and what we can do in practice. So the ideal experiment is often going to be something like randomize representative sample of newborn babies to different households that are fully representative of the population. But in practice, nobody does that, so we do something like we study adoptees, perhaps. And even when with children separated in different children reared apart, we might worry that there is some non-randomness in how they're assigned to environments. Okay. Okay. So I'm going to proceed in a slightly funny way by just giving you I want to just start by focusing on this thing here, on the genetic correlation, and I'll be doing so in order to sort of make the point that if you formulate a model where the only source of resemblance between any pair of relatives is due to their genetic relatedness, you can often explain the data pretty well, but I start there to kind of make the point, and it's something you'll hear me emphasize many times. There's a crucial difference between explaining the data with the model, finding some sets of parameters that fit some moments in the data that you're interested in, and actually having come up with the correct explanation. Okay. So you can try to explain certain features of the data by just assuming that relatives don't share their environments and that there's no gene-environment covariance, and you can see what happens. And that's a very different thing from saying that you found the right explanation. And so then we'll proceed by looking. We'll look at some of the things that go wrong when you try this purely genetic approach. But in many ways, you can think of behavior genetics as sort of taking a model that Fisher formulated in 1918 and trying to fix its deficiencies. Well, first of all, spending 40 years trying to figure out what on earth he was trying to say, and then once that has been established, sort of figure out how do we deal with the obvious inadequacies of the model if you want to apply it to sort of social science outcomes. Okay, and just to preview the conclusion a little bit, I mean in what's called Fisher's classical biometrical model, it's just assumed. So it's assumed that any resemblance between relatives, so relatives don't share any environmental influences. So in other words, these environmental influences are uncorrelated for all relatives, except for one type of relative. I'm curious if anyone can figure out what it is. And there's no gene-environment covariance. So, and there are other, there are other ways. There are certainly ways to modify the model to deal with some of those problems, but usually that comes at the cost of introducing other issues. Okay, so let's think about the rho k g term, and we're going to do so first under some simplifying assumptions. And the most important, there are a lot of them, but the most important are random mating and a bunch of unlinked loci of equal effect. And so Dan already talked about the fact that the genetic factor, right? It has this additive component, it has a dominance component, but in principle you can keep going as long as you want with interactions until you run out of SNPs to interact. And so you can imagine, epistatic interactions between the additive effects of the individual loci, between the dominance deviations, and between the additive and the dominance additive effects and the dominance deviations. And you can keep going like this, okay? And even though neither, as I understand it, neither Fisher nor Wright figured out, even under random mating, the associated with each of these is a variance component. Okay, so this would be the variance explained by all three-way interactions between SNPs at different loci. Huh? Okay, so under some, so it turns out that two people sort of independently. So Fisher and Wright both got stuck. Even with random mating, they were able to infer the genetic covariance for some of these terms, but not the higher order terms. But it turns out you can do it in general. So here I'm just showing some numbers from the textbook. So each number here is the covariance between parent and offspring for the additive genetic effect. This would be the two-way interaction, epistatic effect between additive effects of different loci and so on. And you can keep going with the three-way interactions and the four-way interactions, and you can get some numbers. And it turns out that there's a simple analytic formula that you can use to plug in to calculate what these coefficients should be. So in a model with random mating, you could in principle use this sort of framework to see how well you can explain the data. And the original derivation of this, the generalization of what Fisher and Wright did, was independently derived by two people. I think they published results in the same year, but the more famous one is by Kempthorne, and my understanding is that his derivation is a bit more lucid and easy to follow. But they got through the same answer through very different routes. Okay. So, okay, and it turns out these numbers, some of them you derive from the problem set just from with brute force or statistics, and it turns out that there's a shortcut that you can use. The formula that Kempthorne derived, you can just you plug in some relationship coefficients, and you get the covariance between the specific type of relatives that you're interested in. Okay, so that's a kind of shortcut, and I just want to say something about that briefly, because once you've done it brute force, it's perhaps easier to see why why this alternative approach is equivalent and not simpler. So to learn about the shortcut, we need to define what it means. We need to define the concept of identity by descent. And the definition that we'll be working with is if two individuals share an allele, and you can trace it to the same copy of the allele that was inherited from a common ancestor, then the shared allele is said to be identical by descent. So here's an example. So mother-child or father-child pair, they share exactly one allele identically by descent in a randomly mating population without inbreeding and this stuff. For full, and that's because I got exactly one maternal allele from my. I got exactly one of my alleles. Some locus was transmitted from my mother, my biological mother. Exactly one from my father. So mother-child pairs are a little unusual in that there's no variation around the expectation. The IBD sharing is just one at each locus. Full siblings, on the other hand, so imagine me and my brother. There's some chance that there's a 50% chance we both got the same maternal allele. There's a 50% chance we both got the same paternal allele. And these events are independent, so with 25% of the time we share 0 alleles IBD. I got one maternal allele, my brother got the other one. I got one paternal allele, and my brother got the other one. Sometimes, half the time, we share 1, and 25% of the time we're going to share 2. So it turns out that this IBD, that you can find that the sort of coefficients that you plug into Kempthorne's formula are just functions of this IBD sharing for the different types of relatives. So that's how you, so here's an example, and I've just limited to the first two terms, so I'm excluding epistatic interactions here. But the formula is completely general, so it works just fine. So for a relative of type K, you can calculate the share of the additive genetic variance shared as half times the probability of one allele IBD, given that I'm of type K, and plus the probability of sharing 2 alleles IBD. For dominance, it's just the probability that of share 2 alleles IBD. Okay? So rather than derive these genetic covariances like you did in the problem set from first principles, it's often easier to figure it out from an IBD calculation. I'll try to give some intuition for why this works in practice later on. But first, 2 examples. So let's look at half-sibs. So half-sibs in a randomly mating population, well, they get exactly one allele from a common parent, and they get exactly one allele from different parents or unrelated, and even if the alleles happen to be the same, then by assumption they're not from the same common ancestor. They're just identical in state. So the allele from the common parent is IBD with probability of half. So therefore, it follows that with probability of half, the IBD sharing is 0, and with probability half is 1, and it's never 2, and that's important for the dominance calculation. So we can just figure out what the row half siblings, the additive part should be, and it turns out to be a quarter, and we can do the same for the dominance component, and it's equal to 0 because the half siblings never share 2 alleles IBD. Does anybody have some intuition for why why the dominance sharing is all about sharing 2 alleles IBD? So one way to think about dominance, right, is within locus interaction, and so for the interaction, what matters is the presence of both alleles. So that's half sibs, and we can do the same exercise for full sibs. Same thought experiment, and we get that a quarter of the time we draw exactly the same maternal and paternal alleles. A quarter of the time we draw none of them are shared. A quarter of the time both are shared, and half of the time one of them is shared. Okay, so substituting it into our formula, we get a coefficient of a half for the additive genetic variance and a quarter for the dominance. And I think you did this too on the problem set, if I'm not mistaken. So if nothing, this is a way to kind of check your calculations. So that's where these numbers come from. Now, for some intuition of why it works, imagine some relative pairing of type K, right? Well, the IBD sharing could be 0, 1, or 2. If it's 2, they're like monozygotic twins. They got exactly the same dominance deviation, so those are going to correlate perfectly. And of course, they got the same additive genetic variance. They share the same fraction of the additive genetic variance, also 1. If it's a 1, then they're just like the parent-offspring pairing. And if it's 0, it means that they're conventionally unrelated. So it turns out that for any pairing of relatives of type K, all you need to know is the probability of 0 alleles shared, that they share 0 alleles identical by descent, 1 and 2, and then you just calculate a weighted average, depending on what those 3 probabilities are, where in the first term, what you substitute in is the sharing that you would get for so for when the 2 alleles are shared identically by descent, you substitute in what you get from monozygotic twins. That's the state of the world in which our similarity is the same as a monozygotic twin. When it's 1, you substitute in the parent-offspring coefficients, and when it's 0, you just substitute in the coefficients for 2 unrelated individuals, which are 0. So that's a way to think about it. Okay. So, yeah.
Why, if for 2 unrelated individuals, why doesn't this probability depend on the prevalence of that allele in the population?
For 2 unrelated individuals, what? For 2 unrelated individuals, why doesn't it depend on?
Can you repeat the question, David?
Oh, so the question, as I understood it, was for 2 unrelated individuals, why doesn't it depend on the allele frequency? So I'm not sure how to answer that. I think 2 unrelated people. So.
I guess to rephrase, like what you might expect by chance to depend on what underlying probabilities are, right? Like
So if you take 2 unrelated individuals, right? The allele frequency won't matter because my so we're descended from completely different ancestors. Okay, so my draw in the genetic lottery is completely independent of yours, and the covariance between these 2 draws is going to be 0, irrespective of the allele frequency. I don't know how to. I'm not sure if I'm just restating your question or if I'm.
no, I guess like I was thinking like say for this nephew, like this, say the frequency of the minor allele frequency is like 0.1. So I would have most people going through that.
But remember, when you calculate the covariance, you demean it, right? So you demean the variable.
Oh, I
So
see. Okay.
yeah, I'm not sure if I. Yeah. Raymond.
We also want the distinction between identical by descent versus identical
By state, yeah. No, so that's true, yeah. So it's true that we're. Yeah, it's true that if the minor allele frequency is really low, then there's a very high chance that you and I have the same, that we're homozygous for the other one, both of us. But that doesn't mean that our that's different from saying that our that there's any covariance between our additive genetic effects. Yeah. So I'm, when I'm, so yeah.
Okay. Maybe yeah.
Okay. So.
Thank you.
So to see how far you can get with this kind of model, people usually start with a seminal paper by Fisher in 1918, which is the original form, I think, is indigestible and very hard to understand. It took decades to kind of fix all the errors and clarify what was an approximation and what wasn't, but it was still a remarkable paper. And what I mean, it did so many. It has to introduce all these new concepts in order to ask the question that it set out to address. Yeah, for example, I think it was the first paper to use the term variance, and then he came up with this proposal for how to decompose the genotypic value into different components. So dominance, additive, and then these epistatic interactions in the multi-locus case. So you did that decomposition effectively on the problem set. And what Fisher did was he took the single locus model and he generalized it to multiple loci. And it turns out that many of the insights from the single locus analysis go through, nonetheless. So for example, so the big question at the time was there was a debate between so Mendelian genetics was sort of understood to work for discretely inherited traits. And the big question was, do the same principles explain the inheritance of quantitative traits? And there was a school of thought. I mean, there's a group of people referred to as biometricians, or that didn't think this was the case. And Fisher basically showed that the same principles worked and to reconcile the two schools of thought. And his basic, one part of the argument was if you have to have a lot of loci contributing to some outcome, you add them all up, you quickly get something that looks approximately bell-shaped. Okay. But then he also had to show things like that in equilibrium you get that the variance doesn't all disappear with the passage of time, and these sorts of things. Okay, and his there are many versions of the model, but what's called typically the classic biometrical model, he said that my Y is the additive genetic effect, a dominance, a component, and the residual. Okay. And then he made a bunch of assumptions, and they boiled, and some of them turned out to be quite strong, and that's kind of the point I want to make here. But the basic result was, the basic conclusion was still, the paper did show that you don't need any other rules for inheritance or any other basic principles to explain the inheritance of things like height or other quantitative traits. And so let me show you some data that Fisher sought out to explain. So the data from Pearson and Lee, and these are just correlation coefficients. So this is for parent and offspring, 0.506 for height. This is for full SIB, so there they got a 0.534 correlation. And then for mates, by which he means just husband-wife pairings. So now my first question, so these numbers are from the Lynch and Walsh textbook, and what I'm giving are the averages. So they looked at the parent offspring separately. So they look at mother, daughter, mother, son, father, daughter, father, son, and they found that there is basically these are statistically indistinguishable. So why is that? And that's that's interesting in and of itself. I suppose a priori you could have. It's not obvious that there's this symmetry, but that's a symmetry that's built into the model, and so that's in and of itself that's consistent with the basic framework. Now and the same is true for full siblings. So they looked separately at brother pairs, sister pairs, and opposite sex sibs, and the coefficients were the correlations were again strikingly similar. And then they got a spousal correlation of 0.28. So let me ask you, how long do you think it took to calculate? How many weeks do you think it took to calculate these correlation coefficients according to the textbook? How many weeks? Anybody? Yeah. 8 weeks. Who wants the over and who wants the under? Yeah, I mean, I tricked you a little bit by asking you to answer in weeks. It turns out it took 2 years, according to the textbook. Yeah, go ahead.
With a spousal correlation at 0.28,
Yeah.
wouldn't we expect a higher full SIB correlation then?
Depends on the model, I think. Yeah.
I thought this is the observed.
These are the observed correlations, yeah.
Right. I'm saying, wouldn't we observe a higher full SIB correlation than 0.53 with that degree of assortative mating?
So, not necessarily. I mean, it depends on your theory of what generates these correlations. But and it turns out actually that you can use an additive model only and fit the data pretty well. I'll show that on the next slide. But the point I want to make here is just that with random mating, we run into a problem immediately if we're trying to explain the data. And it's this row here, right? So we can come up with some value of H2A and H2D. There's a variance due to dominance deviations and variance from additive effects that match these moments pretty well. But this is going to be a problem because the predicted correlation is 0. Okay, so Fisher realized this, and he augmented his model to allow for a certain type of assortative mating that I think somebody will talk about later this week. I have put a bunch of bonus slides with technical details at the end. But the key thing I want to convey, I want to make a couple of points just about the assortative mating. So RG is the correlation between the additive factors of husband and wife, mother and father. And it turns out that under a specific assortment scheme that Fisher proposed, you get a restriction on the parameters, namely that this has to be equal to the spousal correlation. Okay? And this is all in equilibrium for those who are wondering. So, and so to Dalton's question, so now we have the same data, but we have a different set of moment conditions. And the answer is, can we explain the data now? They have a kind of ad hoc procedure to answering this question in the textbook that I thought was kind of instructive. So they said, well, let's start here. We know that this is equal to 0.28. So let's substitute 0.28 into there and figure out what the implied value of H2A must be to match this. And the answer is 0.791. And then we can substitute in our 0.791. We can back out the implied value of RG, and we can substitute that in here, we get a number. And that number, if I remember correctly, was like 0.49 or something like that. Okay? So the textbook tells you that that an additive model fits the data. And there's a question, and again, the fact that it fits the data doesn't mean that it's the right explanation, and that's a thing you'll probably hear me say again. There's one more point I want to make, which is that sometimes these this is a free parameter model, and basically you can parameterize it either in terms of spousal genetic correlation, this H2A and H2D, or you can get rid of, say, RG and substitute in the spousal phenotypic correlation instead. But there's a restriction that the logic of the model requires to hold that RG over H2A has to be equal to the spousal phenotypic correlation. And so if you don't impose that restriction in estimation, you're not really estimating the model. Okay. Now let's go back here and just show one more issue. Maybe this is what Dalton was getting at, and I didn't understand it. But if you just look at this, you also get something funny. You get a predicted, narrow heritability of a little bit over 100%. Maybe is that what you were alluding to, or yeah. Okay. Sorry, I didn't realize that. So you get strange answers without the assortative mating. Fisher's solution was to add the assortative mating, and then appeal to dominance whenever the assortative mating augmented a model didn't deliver what he wanted. And so let's think about this. So in general, when you look at behavior genetic explanations, I think it's a good practice to think about what are the core underlying assumptions. Okay, so in the Fisherian model that we just looked at, for example, there's some very, very strong but subtle assumptions that go into the assortment scheme that's being conjectured. And the reason Fisher had to make these assumptions is that he could observe the spousal correlation, but not these other things. So he had to make some assumptions in order to back out the unobservables from the spousal phenotypic correlation. And the only way to do that, or his preferred way of doing that, was to write down a model where the assortment is what's called on phenotype. And assortment on phenotype just means that any correlation between one of the factors that determine my phenotype, so my A, my D, or my E, and that of my spouse, vanishes if I have control for the spousal phenotype. So that's what people mean when they say sorting on phenotype. Okay, so the random mating benchmark doesn't work very well. The augmented model with phenotypic assortative mating, you can pick parameters that roughly match the moment in the data. Later on, we'll talk a lot more about how you pick, how you do this estimation more formally. But for now, I think the kind of ad hoc approach is probably fine. Okay. So now back to evaluating this sort of stuff. I think when you look at behavior genetic explanations, it's important to think about the core structural equations of the model and what's being assumed. And they tend to embed important assumptions, sometimes very indirectly and opaquely, about processes, not just biological processes, but also psychological, sociological, economic processes that social scientists often have interesting things to comment on. Being just transparent about what these assumptions are, I think, can be useful for forcing clear thinking, but also for thinking about possible biases and worrying about how to detect them and what are the data we could maybe incorporate to address them. And these assumptions easily get lost when there's too much attention to discussions about fit diagnostics and whether a particular model fits the data. That's a statement you often hear. And a general principle that I think is very important to remember is that if your model is misspecified, you're almost certainly going to get biased estimates, but there's no guarantee that it leads to a bad fit. Okay? So this is an example of a model that fits the data pretty well, but we probably don't think that all gene action for height is additive, and that we don't need anything else to any other forces in the model to explain data. Or maybe it's fine for height, but we worry that this kind of model, if applied to other social sciencey phenotypes, might give you a good fit but terribly misleading explanations. Okay, so what are these kind of core assumptions to think about here? Well, the main one that I've already alluded to is that the classic model just rules out correlation between the environments of relatives. So remember and also gene-environment covariance. So remember when I said we're going to write the phenotypic correlation of kinship K as a function of these three things: a genetic term, an environmental covariance, and a gene-environment covariance. The Fisherian approach is just to ignore the last two terms and see if we can explain the data with just a genetic resemblance parameter. Now, and you can think of a lot of modern behavior genetics as trying to kind of tack on environmental resemblance to this scheme in a coherent and consistent way. Okay, now this feature of the model that basically rules out correlation between relatives, there's one exception that we'll get to. It seems at odds with common sense and just plenty of evidence. I'll show you some. It limits the scope for credible applications of the model, and as I said, there's one exception to the rule, and it's not for siblings or something like that. It's for spouses. So here's another fun Goldberger quote on what this means. Like, and this is something that's very hard, I think, to detect unless you really go through the nitty-gritty details of the matrix algebra. So what it means is that we're assuming an implication of the assumption of phenotypic assortment is that we end up with a model in which my wife's, from the point of view of the husband, childhood environment was similar to mine, but not her brother's, nor indeed to her children's. And he points out this makes the model not only implausible, empirically inadequate. Okay, and so people have worked very hard to try to address these kind of issues, and I think Chapter 7 in Lynch and Walsh has a good review on work by, I think, Cloninger and Rao has some good stuff on this in the 70s. And the way they solved it was basically to assume additive gene action, but that, you know, so ignore all the non-additive stuff. But then that did allow them to formulate a coherent model with sort of analytical solutions that allowed for gene environment covariance and resemblance between relatives and these sorts of things. Did somebody have a comment? No? Okay, so why did I say that it's empirically inadequate? And I want to show you just some data from, so this is data from conscription, so it's all for brothers. It's Swedish data that I was involved in collecting. And it's showing you for 7 different types of relatives, so each of these you can think of as a K, the phenotypic correlation. So we have so the twins are all reared together. So we have monozygotic twins reared together, dizygotic twins reared together, full sibs reared together, full sibs reared apart, half sibs reared together, half sibs reared apart, and adoptees. So a couple of things stand out, and I just want to point them out to you. So the first is that the similarity among siblings, types of siblings who are reared together, is increasing in their genetic relatedness. So if we start with adoptees, we generally get fairly low numbers. I should say, yeah, this is a score on a cognitive test, and this is a military psychologist's evaluation of your ability to deal with wartime stress, technically. But in practice, it turns out that this evaluation predicts all kinds of stuff, all kinds of life outcomes. So you can think of it as a socio-emotional skill if you don't like the label non-cognitive. So adoptees first. Then we go to half siblings, which should be more related than adoptees. We see consistent pattern of greater resemblance for, I think, all the traits. Yep. There's one, I guess socio-emotional is a tie, but there's still a fairly clear pattern. Now go to DZ twins, in greater similarity, and the MZ twins, even greater similarity. I mean, so there's a clear systematic pattern in the data. Among siblings reared together, those who are more genetically related turn out to be more similar. And then we have stylized factor number 2, which is that siblings reared together are more similar than siblings reared apart. And this is the feature of the data that the Fisherian model can't explain, right? Because by assumption it says if 2 people are brothers, their similarity will be the same regardless, because the only source of similarity between them is their shared genes. And that doesn't appear to be true in the data, right? We see again consistent pattern of greater similarity for siblings reared together than siblings reared apart. And if you're wondering how is this defined, well, every 5 years in the data there's a census, so you can see which house somebody was domiciled in, and that basically allows you to if a child was domiciled in the same house as his or her parents every census between the ages of 0 and 18, then we say that this child was reared by his or her parents, and if 2 siblings were reared in the same households, then we say they were reared together. Okay, we can do the same for half sibs. We see a similar pattern, and this is the second striking feature, the striking thing when you look at these data. So to think about how you might go about quantifying, separately identifying, these effects of this shared environment from shared genes, it's useful to consider thought experiments. And so here's one thought experiment. We'll go through a couple and just talk about them.
David, we should stop soon.
Oh, I'm running over. Okay, maybe I'll just resume this tomorrow then. That's fine. I didn't realize I'd taken up so much time. Sorry.
No.
Okay, let's pause there then, and I'll this will resume tomorrow.
I have a quick question.
Go
about
for it.
something you said. You said that the Fisher's model was allowing for correlation of spouses environments.
Yes.
Did that show up in the earlier slide you showed with the Lynch and Walsh model?
Was that the RNC?
Are you referring to this? Yeah. That's a good question. So I think the answer is that yeah, you can't see it from this way of writing the moment conditions, but you can have some notes on this. You can reparameterize it so that it's more clear. And the explanation is, I think, that one of these terms can be written as the as a function of, like, rewritten as a function of other parameters where it shows up. But it's a good question.
What is RZ?
RZ, where do you see RZ?
Z. Like
Oh, G. RG is the correlation between the additive genetic factors. Yeah? And RY is the spousal correlation. So RY has to be equal to this. Yeah.
the additive factors of the spouses.
Yes. Yeah. The AXs. Yeah. Okay. I mean, I'll stop there. I'll have a better question to answer your question tomorrow. It's a good one. Yeah.
Okay, so let's take a 5